Everyone, we have already studied section 2 of paper CHA 5304, and in section 2 various chapters are there as a chemiluminescence, fluorescence, phosphorescence, and last one is a NMR. In NMR, H1 NMR, carbon 13 NMR and the 2D NMR is there. We have already started the H1 NMR, the basics of the NMR spectroscopy, then the coupling constant, chemical shift, then energy associated with the nucleus and the magnetic field. We have already solved two problems based on a relaxation and the energy between two states. As one state contains plus half spin and second excited state contains minus half. The energy gap between these two energy levels is delta E, and we have already calculated this. And the problems we have seen how the signal is obtained, the H1 NMR spectroscopy in which the hydrogens are important. The neighboring carbon atom contains number of hydrogens and the peak observed is n plus 1. Number of hydrogen plus 1. Such types of peaks are observed in the NMR spectroscopy: singlet, doublet, triplet, quartet, and so on. Then multiplet is there. So such a type of peaks are observed in the NMR spectroscopy. So to study the number of hydrogens, the structure of any compound, or a chemical environment of any molecule or any atom, this can be studied by NMR instrumentation. So we are going to learn NMR instrumentation. We know the basics of NMR, but how the NMR spectroscopy works, this can be studied in this lecture. So see, this is a schematic diagram of NMR spectrophotometer or a spectrometer. See, we have already studied this diagram. Again, I am telling this. See, this is the instrumentation of a NMR spectrometer. Initially, you have to dissolve a sample in a solvent. Usually, CdCl3, that is a deuterochloroform, is used as a solvent. So see, first step is the sample is dissolved in the solvent in this NMR tube. So here is the NMR tube. See, this red color is a NMR tube in which solvent is kept, and you have to add a solid into it. So it becomes a solution, and the solvent used is a CdCl3 for dissolution of a sample. So this is the first step. So solvent is inserted. Then here is a superconducting magnet in which the magnetic field is applied. Then see, the view diagram of a sample tube is like this. Sample tube. See, this is the red colored component in which we can going to put a sample. See, its view is like this. This is the sample tube. A capillary-like structure is seen in this diagram, and this is a sample tube in which the sample is kept. And this sample is kept in the magnetic field. A radio frequency generator then irradiates the sample with a short pulse of radiation. So see, in the NMR probe, the sample is rotated in the magnetic field and irradiated with a short pulse of RF radiations. Means see, our sample is kept inside the probe and there is a magnetic field. So superconducting magnets are used for the applying of the magnetic field, and there is a radio frequency generator, that means a RF source or a light source which emits number of radiations, and then there is a resonance occur. The ground state nuclei goes to the excited state and spins get inverted. When the nuclei fall back to the lower energy state from the excited state, the detector measures energy and a spectrum is recorded. The superconducting magnets in modern NMR spectrometers have coils that are cooled in a liquid helium and conduct electricity with essentially no resistance. So see, these superconducting magnets, here the superconducting magnets are there, and nowadays this superconducting magnets in NMR instrumentations have a coil like structure, and that coils are cooled in a liquid helium, and thus coils can conduct electricity with no resistance. So this is a schematic diagram of a NMR spectrometer. Now we are going to see a block diagram of the same instrument. See, this is a block diagram. See, here is a first radio frequency transmitter, means a RF source. Then here is a radio frequency amplifier, means signal is enhanced. Then there is a detector, then audio amplifier, and lastly we can get a spectrum. And here is an oscilloscope or a recorder. So see, this is a instrumentation. See, this one is our sample. Here two magnets are kept. These magnets are a superconducting magnet. This is a magnet. So see, same diagram is here. This diagram is from our book and this is from the net. And here our sample is kept inside the probe. There is a magnetic field applied in the form of coils, as I said nowadays. And with magnetic field there is a radio frequency light or an emitter that emits light. This light can be absorbed by the sample molecules. Then these electrons or protons goes to the excited state. In the excited state, spin get inverted, then it comes to the ground state with the emission of a light. The emitted light goes to the radio frequency amplifier. The signal get enhanced. This goes to the detector and then comes to the audio amplifier. This generates a spectrum on a recorder. So this is the block diagram of a NMR instrumentation. Same diagram is here. See, magnetic field sweep is here. Then again magnetic pole is here. So see, our sample is kept here in between. Here written is a sail, and this sail is rotating continuously. And the magnetic poles are there, north pole and the south pole, with the magnetic field base 0. And this is a receiver coil. This is called as inner coil again. Here is a sweep coil. The coil around the magnet is the sweep coil. This we have already seen. The magnets are the coil-like structures because coils conduct electricity with no resistance. So the coils are there, and these coils are called as the sweep coils. Then see, sample is here. The magnets are kept here. Then magnetic field is either increased or decreased, or it can be controlled by using the magnetic field sweep. Then radio frequency transmitter is there. Here radio frequency receiver is there, and the readout device. Same diagram is as previous diagram. Sample, around the sample there is a coil and the sail is continuously rotating. The magnetic field is applied and the radio frequency transmitter is there. Radio frequency transmitter emits light. This light can be absorbed by our sample molecules or the sample protons, and then this signal get transfers toward the radio frequency receiver and then to the readout device. So this is the diagram of NMR spectrometer, and the arrows on the sweep coils indicates the direction. This arrow shows the direction of current flow through the coil so that the applied magnetic field is in the indicated direction. So current flow through the coils and the applied magnetic field is indicated the direction. So see, initially diagram starts from here. Magnetic field sweep. The direction of the magnetic field is in this way. the current is passed, then it comes to the north pole, then north pole to the cell, from cell it passes to the magnetic pole which is the south pole, then signal is again get reverted to the magnetic field sweep, then goes to the detector. This is the first way by which the signal is obtained. Or another way is here, the magnetic field sweep to the north pole to the cell to the readout amplifier or the radio frequency receiver, then goes to the readout device. So two-way analysis can be done. One time the magnetic field is kept constant and the radio frequency can be measured on the instrument or a readout device. Or a second condition is there, the radio frequency kept constant and the magnetic field is changed. At that time the detection is done on the instrument. Means there are two types of fields. First one is a magnetic field and second one is a radio frequency light is used, that is a electric field is there. Means electric field and the magnetic field, both types of fields are there. You have to keep at least one field constant. Keep magnetic field constant, radio frequency can be measured. When radio frequency kept constant, you can measure the magnetic field. So here at the readout device, two ways are there. See, first way is a X way and second way is a Y way. So two types of measurement is done. See here, if radio frequency transmitter frequency is kept constant, at that time you can measure the magnetic field by this way, and when the magnetic field is kept constant and radio frequency is measured on the detector. So this is the actual working of a NMR spectrometer. So see, components in NMR spectrometer: an intense, homogeneous and stable magnetic field, then a probe which enables the coils used to excite and detect the signal to be placed close to the sample. So this is called as a probe. This circular coil is called as a probe and this is kept near to the sample because it enables the coils used to excite the proton and detect the signal to be placed close at the sample. Then a high power radio frequency transmitter capable of delivering sharp pulses. Then a sensitive receiver to amplify the NMR signal. Then a detector to convert the NMR signals into a form of a data which is stored in a computer memory. And a pulse programmer is used to produce precisely timed pulses and delays. Means pulse programmer measures the light which passes for a particular time. Then a computer to control everything and to process the data. Means each component is used for a particular use. Firstly, a stable magnetic field used, which is an intense and a homogeneous magnetic field. Then probe is used to excite and detect the signal and it is placed close to the sample. Then the radio frequency transmitter which provides a high power. Then a receiver which amplifies the NMR signals. Detector converts NMR signals into a data in a computer memory. Then a pulse programmer which produces precisely timed pulses and delays. Then a programmer to control everything and to process the data. So these are the components of NMR. Then magnet must be strong, stable and produce a homogeneous field. Homogeneous means the field does not vary in strength or direction from point to point over the space occupied by the sample. Means there is no change in strength of a magnetic field or a direction from point to point. Then range of the magnetic field is from 60 megahertz, that is 1.4 Tesla, to 700 megahertz, that means 16.4 Tesla, and higher, means range is 60 megahertz to 700 megahertz. And there are two parts of magnets, superconducting magnets and Shim coils. So two types of the magnets are used. Then a superconducting magnet. See, this is our sample tube, and around this there is a coil. This is of a magnet. So it is made up of superconducting NB or tin or SB or Ti wires. The magnet is submerged liquid helium. See, this is a helium, and in which the magnet is submerged for providing the sufficient cooling. It provides cooling, and the magnet and the liquid helium reservoir are encased in a liquid nitrogen. Means see, outside that there is a liquid nitrogen. In that the magnet is kept and the liquid helium is kept. So this is for to decrease the evaporative loss of more expensive liquid helium. So helium is more expensive, that's why it kept in a nitrogen reservoir, and inside it there is a magnet or a superconducting magnet. This is kept for to decrease the evaporative loss of a helium, which is more expensive. Then the sample probe is put in the bore along with a set of room temperature Shim coils. So sample probe, see, this one is a insert sample. Here we insert sample from here. Here is a vacuum chamber, then there is a magnet, here is a liquid helium, then outside that there is a nitrogen, and we have to insert a probe here. Then Shim coils, either superconducting magnets are used and made up of NB, tin, SB and Ti. Then Shim coils, see, these are the Shim coils, this diagram. And the Shim coils are used for making magnetic field homogeneous. Means simply Shim coils converts the field into a homogeneous form. Through these coils, current is adjusted until the magnetic field has a required homogeneity. Then Shim coils produces a magnetic field. Then the small residual inhomogeneities in the main magnetic field, and naming of Shim coils is done on the basis of their direction, which they generate the corresponding magnetic field. Then the sample holder, see, we have seen a capillary like structure in this diagram. See here, we have seen this capillary like structure. This is called as a sample tube, and we can say same as a sample holder. So the sample holder in NMR is normally a tube shaped and is therefore called the sample tube. This is a transparent material, and this is for the radio frequency light absorption. This tube is durable and chemically inert. Means this does not react with the sample, and made up of a glass or pyrex material. Then these are sturdy, practical and cheap. Means anyone can buy for the NMR instrumentation easily. They are usually about 6 to 7 inch long and 1 by 8 inch in diameter, with a plastic cap. This cap is made up of a plastic material, and this type of tube is used for obtaining spectra of a bulk samples and solutions. So this is a sample holder or a sample tube made up of glass or a pyrex material. Its length is about 6 to 7 inch and its diameter is 1/8 in side. Then a probe. So we have kept this sample tube inside the probe. See, this sample tube is kept inside this probe. So the probe is like this. This is the structure of a probe. Actually, this one is a probe. This is a exact view of a probe. From this hole, we can put the sample tube inside the probe. So, see, such way we can put a sample tube or a sample holder inside the probe. So, see, this is a probe. Probe is a sample holder or probe in which the sample holder is placed. It contains an air turbine to spin the sample holder. Means we have shown the diagram in a circular motion, or a tube is continuously in the rotating motion. See, this arrow shows the tube is continuously in the rotating motion, or it rotates in a circular motion. This is due to a probe and used to excite and detect the magnetization in a radio frequency of sample. The most essential component is the RF transmission and the receiving coil. Means, as we have already written, the coils are kept near to the sample, and this is for the rotation or the exact transition of a proton from ground state to the excited state, and the emitted lights can be measured. So, for a maximum sensitivity, a fixed frequency probe is needed, and this is required for each nucleus like H1, carbon 13 and fluorine 19. So see, a probe means what? A probe in which the sample holder is kept. It contains an air turbine to spin the sample holder, means there is a rotation to the sample holder or a sample tube due to the probe while the spectrum is collected. Then it used to excite and detect the magnetization in radio frequency of a sample. Then this is used for maximum sensitivity, and a fixed frequency probe is required for H1, carbon 13 and fluorine 19. This is a exact view of a probe. From here we can put a sample holder in it. It has a continuously rotating motion. Due to that, the sample can easily excite. Then RF generator and the detector. RF means radio frequency generator, and the source is the RF crystal oscillator, which is used for the radio frequency light source. The output of the oscillator is amplified, mixed and filtered to produce monochromatic RF radiations, and it can be delivered to the sample. So see, RF crystal oscillator, it converts the polychromatic light into a monochromatic light or a monochromatic radio frequency light, and this light can be transferred toward the sample, and this pulse is of a 500 megahertz frequency. So this is a RF generator or a radio frequency generator. Then the radio frequency detector is used here. The NMR signal emanating from the probe is detected by a digitizer receiver at regular time intervals. So NMR signal which is emitted from the probe or a sample, it can be detected by a digitizer or a digital receiver at a regular time interval. Time is fixed as we have written. The pulse time is needed, or a delayed pulse time can be measured. These signals in the time domain must be converted to a frequency domain by application of a Fourier transform spectroscopy or other mathematical transformation. So see, Fourier transform spectroscopy FID shows such types of plots as a wavy diagram is here with respect to time, and this can be converted into a spectrum, and this is in the form of a frequency domain, and this is the exact diagram of a NMR spectrum. So sample preparation is written here. So samples are degassed to remove oxygen, means oxygen is removed, and filtered to remove iron particles. Both O2 and iron are paramagnetic and cause undesired line broadening. So see, for sample preparation we have to remove oxygen and the iron particles. So oxygen and ions are paramagnetic and cause undesired broadening, means peaks are not in the form of a line but they will be in the form of a continuum. So to avoid that we have to initially degas, means there is a removal of oxygen and iron particles. Then liquid samples. Neat non-viscous liquids are run as is by placing about 0.5 ml of the liquid in a glass NMR tube. So a clear solution is required for the analysis and its volume is needed 0.5 ml. This can be added into a sample tube. Then liquids can be mixed in a suitable solvent. Concentration is kept generally 2 to 10%, and solvent is mainly CdCl3. Then for the solid samples are initially dissolved in a suitable solvent, and then this can be used for the analysis. Then the sample size which is required is around 2 to 3 milligram, which is dissolved in a 0.5 ml of a solvent. So see how much sample is required, only 0.5 ml. This will be either liquid or solid, it is all in a liquid. The sample required is 0.5 ml. And for the gaseous samples, there is no sufficient sensitivity to analyze gas phase samples, and gases must be concentrated by absorbed in a suitable solvent. So NMR is generally useful for liquid and the solid samples, and it can not give sufficient amount of sensitivity for the gas samples. And if we are going to study the gas samples, at that time we have to concentrate gases, and then these gases can be absorbed in a suitable solvent, and then only we can learn a NMR spectrum. Then the solvents. A suitable solvent for NMR should meet the following requirements. The solvent should be chemically inert toward the sample and the sample holder. Then sample have no NMR absorption spectrum itself for a very simple spectrum. Means sample does not show absorption spectrum, or if it shows it will be very simple spectrum. And third point is easily sample is recovered by distillation. For example, if the original sample is required for other testing, means there is no loss of sample. And the base solvents for proton NMR contains no proton, means the solvent in which proton is not present or hydrogen is not present, this is used as a base solvent. And the deuterated chloroform, that is a CdCl3, deuterated water D2O, and many other deuterated solvents are used in the NMR spectrometer for the dissolution of a solid. Then advantage of NMR spectrometer is one of organic compound can be determined quantitatively in the presence of a different type, such as the percentage of alcohol in alkene, then amines in alcohols, aromatics and aliphatics in petroleum. This can be analyzed quantitatively. Then NMR can be used to provide determination of chemical purity and quantitative measurements of impurities in material. So this is used for the structure determination, quantitative analysis, and use helpful for finding the purity of a compound or finding impurities. So this is all about the NMR spectroscopy. So our H1 NMR is complete now. We will learn the carbon 13 NMR in next lecture.